Hi. The- this is life science and cell biology in a nutshell. So, the difference between the terms anabolic and catabolic. Anabolic is when um, you are building up um, complex molecules. Anabolism is also called biosynthesis. It is a sequence of enzyme catalyzed reactions. Um, so, where smaller molecules are used to make larger molecules, and usually with the use of ATP or energy or hydrolysis of adenosine triphosphate. This builds up organs, tissues, muscle tissues, etc. So this is anabolic state, and the opposite of this is catabolic, breaking down muscle fibers, not working out and not eating enough, and not getting sufficient rest can lead to catabolism. This so so while anabolism forms complex molecules in living cells from nutrients with relatively simple structures, um, and cellular components are synthesized, such as carbohydrates, proteins, lipids, etc. This requires energy in the form of ATP, or energy-rich compound, produced during catabolism, or the breakdown processes. So in growing cells, anabolic processes dominate over catabolic processes. And in non-growing cells, a balance will exist between the two processes. So the nature and stages of biosynthesis of cellular components. The biosynthesis of cell components, or anabolism, may be regarded as occurring in two main stages. In the first, intermediate compounds of the central routes of metabolism are diverted from further catabolism and are channeled into pathways that usually lead to the formation of the relatively small molecules that serve as the as the building blocks or the precursors of macromolecules. In the second stage of biosynthesis, the building blocks are combined to yield macromolecules such as proteins, nucleic acids, lipids, and polysaccharides that make up the bulk of tissues and cellular components. In organisms with the appropriate genetic capability, for example, all the amino acids can be synthesized from ammonia and intermediates of the main routes of carbohydrate fragmentation and oxidation. Such intermediates act also as precursors for the purines, the pyrimidines, and the pentose sugars that constitute the DNA, and for a number of types of RNA, which are all called nucleic acids, a group. The assembly of proteins necessitates the precise combination of specific amino acids in a highly ordered and controlled manner This in turn involves the copying or transcription into RNA of specific parts of DNA. So nucleic acids and proteins, which lead from the nucleic acids, proteins are made. The first stage of biosynthesis thus requires the specificity normally required for the efficient functioning of sequences of enzyme-catalyzed reactions. The second stage also involves directly for protein and nucleic acid synthesis, less directly for the synthesis of other macromolecules. The maintenance and expression of the biological information that specifies the identity of the cell, tissue, and the organism. Utilization of ATP is the following. The two stages of biosynthesis, the formation of building blocks and their specific assembly into macromolecules, are energy-consuming processes and require ATP. Although the ATP is derived from catabolism does not drive biosynthesis. As explained earlier, the occurrence of chemical reactions in living cells is accompanied by a net decrease in free energy. Although biological growth and development results in the creation of ordered systems from less ordered ones, and of complex systems from simpler ones, these events must occur at the expense of energy-yielding reactions. The overall coupled reactions are, on balance, still accompanied by a decrease in free energy and are thus essentially irreversible in the direction of biosynthesis. The total energy released from ATP, for example, is usually much greater than is needed for a particular biosynthetic step. Thus, many of the reactions involved in biosynthesis release inorganic pyrophosphate rather than phosphate from ATP, and hence yield AMP rather than ADP. Since inorganic pyrophosphate rarely undergoes virtually irreversible hydrolysis to two equivalents of inorganic pyrophosphate, the creation of a new bond in the product of synthesis may be accompanied by the breaking of two high-energy bonds of ATP, although in theory one might have sufficed. The efficient utilization for anabolic processes of ATP and some intermediate compound formed during a catabolic reaction requires the cell to have simultaneously a milieu favorable for both ATP generation and consumption. Catabolism occurs readily only if sufficient ADP is available. Hence, the concentration of ATP is low. On the other hand, biosynthesis requires a high level of ATP and consequently low levels of ADP and AMP. So suitable conditions for the simultaneous function of both processes are met in